Three basic cell types I want you to know for the JGA. So the JGA consists of what? And you should just list three cells. One, these macula densa cells, MD. These are specialized cells in the tubule of the thick ascending limb, macula densa. They sense things in the urinary fluid. We'll talk about that later. So the macula densa is one section. Two are these cells of the afferent and efferent arterioles. Just go over the anatomy again. Here's your AA. That's your afferent arteriole. Okay? The afferent arteriole is bringing blood into these glomerular capillaries. And then plasma, water, and electrolytes actually filter from these capillaries into this Bowman space. BS stands for Bowman space, named after William Bowman, who was an English physician. I think it was at the age of 25 or 26 that he actually discovered filtration of the kidneys. So this is called Bowman space right here, and that is the initial process of urinary formation. So that's where filtration occurs. And then the efferent arteriole carries the rest of the blood back out to the rest of the kidney. And then out here is another capillary bed called the peritubular capillaries. So going back to the juxtaglomerular apparatus, three structures. One is the macula densa, two are the granular cells of the afferent arteriole, and three are these EGM, extraglomerular mesangium. EGM, extraglomerular mesangial cells, right in here. Apparently they have some phagocytic activity, some protective function that they can do and take away some bacteria and engulf them, and they also secrete some things, but some of the function is still not known about those. So JGA, three things you just list: macula densa, extraglomerular mesangial cells, granular cells. Okay. Now going back to here, in the renal corpuscle, the renal corpuscle includes this Bowman space and the glomerular capillaries. Usually I'll just say what happens at the glomerulus, what occurs at the glomerulus, and that's where filtration occurs. But basically it's the renal corpuscle including these glomerular capillaries. It's really like a fist that you can shove into a balloon, and then the balloon kind of covers these capillaries. And that's the way it actually forms during the formation phase of all of these nephrons. In the renal corpuscle, we have these capillaries, mesangial cells. Glomerular mesangial cells are kind of embedded in there. These are irregularly shaped cells that have some phagocytic activity. They secrete a matrix and they degrade a matrix. And so I'll talk a little bit about this later on, but these mesangial cells apparently do serve an important function, both with filtration as well as kind of cleaning up some bad things that might get filtered into the kidney. And we have these podocytes. This is a parietal epithelium with foot processes. P stands for podocytes. So interestingly, what happens is, you know what capillaries are. So they're the endothelial cells, right? Very thin blood vessel. In the kidney, this parietal epithelia actually forms what are called these podocytes that surround these glomerular capillaries. And those podocytes apparently serve a very important function in glomerular filtration. So in order to go from the glomerular capillaries into Bowman space for filtration, you have to go through what's called a filtration barrier. And I want you to know what the filtration barrier is. One, the endothelial cells they got across of the glomerular capillaries. Two, the glomerular basement membrane, which actually includes some of the basement membrane of the endothelial cells as well as the basement membrane of the podocytes. And then three, the podocytes themselves are these so-called filtration slits that we'll talk about. So the filtration barrier, endothelial cells, basement membrane, podocytes. Endothelial cells, basement membrane, and podocytes. This is where a lot of the kidney diseases occur. Okay? So this is why you have to know the structure and the function of the renal corpuscle. Okay? Glomerular nephritis, diabetic nephropathy, hypertension begins to break down this filtration barrier, and bad things can happen. Okay, here's an electron micrograph of this glomerular capillary. Now here's a cell body of a podocyte. These are the capillary cells themselves. So this is what you would see basically if you were in Bowman's space, okay? And you can see it's really a monstrous-like structure of this thing, and it's been difficult to study of just what the function of these podocytes are, but you can see it kind of wraps around in what's called a finger-like projections, and then from those there are actually these other projections that kind of interdigitate right along the glomerular capillary. It's believed that this serves a very important function for selectively filtering what needs to be filtered and what doesn't need to be filtered. Now this here in B is what you would see if you were actually in the capillary itself. Okay, so these are some nice EMs here. Okay, you can see that the endothelial cells are very fenestrated, okay, very leaky. Okay, and then there's some other structures in there as well. So the filtration barrier that we'll talk about quite a bit when we talk about the renal regulation of glomerular filtration rate exists of that. Okay, so the endothelial cells of the capillaries, the basement membrane that things have to go through. Okay. There's actually kind of 3 layers of that basement membrane. And then they have to get through somehow these podocytes. And they apparently, a lot of things get filtered right through these filtration slits. Okay? So I want you to have just a basic understanding of what this looks like. And for test purposes, know those structures of the filtration barrier. Also, this is very electrically negatively charged as well. So there's a lot of glycoproteins along these cells. So you can just visualize it as having a bunch of negative charges all along the filtration barrier. Here's an EM of glomerular capillaries just to show you this. This was, I don't know how they isolated that. It's really difficult to do, but here's an interlobular artery branching off into an afferent artery, into this tuft, so-called, of capillaries, where filtration occurs, and then it branches off into an efferent arteriole, going back into the rest of the kidney, into the cortex of the kidney. So when I begin talking about glomerular filtration tomorrow, I'm going to talk a lot about afferent arteriole, glomerular capillaries, efferent arteriole, peritubular capillaries, okay? When we're talking about filtration, what gets filtered and what doesn't get filtered. If you take a urinalysis, what would be in the urine that would tell you the kidneys are not functioning well? Okay, what's in the plasma that causes the oncotic pressure to be so high? Yeah, the plasma protein, the albumin. So that is the main thing that tends not to be filtered. Of course, red blood cells get into the capillaries too, but they're too big to go through the filtration barrier. So if you're filtering red blood cells, urinating blood, okay, you obviously know that something's wrong with the kidneys. Could be a lower urinary tract infection, but generally the filtration barrier will keep all of the blood cells out. Okay? And the albumin, a little bit of albumin does